This method can help to answer key questions in the field of tuberculosis, such as what is the role of adaptive immune responses and the pathogenesis of this multifactorial disease. The main advantage of this nucleic acid-based method is that both bacterial loads and gene expression levels can be measured from the same individual.
To begin this procedure, culture M. marinum on a 7H10 plate as outlined in the text protocol. Transfer 10 milliliters of 7H9 medium containing ADC enrichment, polysorbate 80, and glycerol to a cell culture flask. Then, use a sterile 1 microliter inoculation loop to aseptically transfer 1 loopful of the M. marinum bacterial mass into the flask. Leave the cap loose or use a filter cap to allow for sufficient gas exchange. And culture at 29 degrees Celsius in the dark without shaking for 3 to 4 days until the OD600 reaches about 0.7. Dilute and continue culturing the bacteria as outlined in the text. To begin preparing the bacterial solution for infection, transfer 1 milliliter of the culture into a fresh tube. Centrifuge at 10,000 G for 3 minutes. Remove the supernatant and resuspend the pellet in 1 milliliter of sterile PBS. Using sterile PBS with 0.3 milligrams per milliliter of phenol red as a tracer, dilute the suspension to reach the desired bacterial concentration. After this, use a 1 milliliter syringe to slowly pull the